Ganesh. Okay. 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 Thank you. So, Dr. Jubair Wasim Sayed is presently working as postdoctoral fellow, Cleveland Clinic, Florida Research and Innovation Center, Port St. Lucie, USA, and he has completed a PhD in CSIR CCMB, Hyderabad, India. He has also hello. So he has worked under a project at CSIR CCMB, Hyderabad. He has completed his master's in science, MSc in microbiology from University College of Science, Usmania University, Hyderabad. He has also completed his BSc in microbiology subject from NES Science College, Nanded. So it is a proud moment for us that he was a student of or alumni of this Science College, Nanded. He has completed his BSc from Science College, Nanded during 2008 to 2010. He has awarded many fellowships and awards. He has awarded an Nobel Council of Scientific and Industrial Research, that is CSR, NET, JRF fellowship with All India rank 39. He has also qualified graduate aptitude test in engineering, that is GATE in life sciences with rank All India rank 348. Awarded Best Poster Prize Award for the presentation of work at CSR Foundation Day successively in 2014, 2016, 2018, held at CSIR CCMB Hyderabad. He has also a very important publication in his credit with high impact factor journals. He has worked as a postdoctoral fellow, as a PhD student, project fellow in various projects funded by various institutes at different places, particularly in CCMB, Hyderabad, and presently he is working as a postdoctoral fellow at Florida Research and Innovation Center, USA. He has also presented his paper in various conferences, poster presentation. He has also took part in the poster presentation, oral presentation at various national and international conferences. He has also takes part in various workshops, particularly imaging and image analysis. The workshop held at Institute for Stem Cells and Regeneration Medicine and C Cellular Molecular Platform campus in June 2020, and various workshop he has attended. So, dear participants, it's our really pleasure to invite Dr. Jubair Wasim Sayed as a chief guest for today's session. I welcome Dr. Jubair on behalf of all the participants and on behalf of Research Committee Science College Nanded for this today's morning session. For your information, this is a pre-PhD workshop. We have conducted this workshop since 20th March 2023 to 13th April. So in total, it is a 21 days PhD workshop. And in that, we have organized various lectures for our research students. These are the newly joined research student, and it is compulsory for them to do their PhD research work. All the research, there are in total 46 research students are present within this workshop, and from different 5 subjects, particularly mathematics, physics, chemistry, botany, and zoology, and 1 international student, which is from mathematics background, is as a research student joined here for this meeting. So I welcome Dr. Jubair sir on behalf of all the participants, and I request him to start his session. Dr. Jubair sir.
Thank you so much for the kind introduction, sir. So I would be presenting my PhD work, which I have done at CSR CCMB. Give me a moment. Yeah. Is my presentation visible now?
Yes, sir. Yes.
Okay. So good morning, everybody. It's almost midnight here, and you might see some weird sounds because there is a thunderstorm going on outside. So it's like a great pleasure to come back to the institute where you studied and perceived and shaped your career towards whatever I was aiming for. So I did my BSc from Science College, and basically that has been a turning point for me to get into research. And then from there, I started taking research as more seriously. And when I got into my master's, I had decided that I will pursue PhD. So at the time itself, I had lots of things figured out, that how am I going to do that? What are the institutions and what are the priorities for me? So when I finished my master's, because of the competitive exams, I got an opportunity to get into CCMB for my PhD. And the area of the work which I was more fascinated was cell biology at the time. And currently I am working towards post-pathogen interaction during my postdoc. But again, it is the signaling involved because of the pathogen. So during my PhD, I worked extensively on how optineurin is associated with several neurodegenerative disorders. So the neurodegenerative disorder, which I'm going to talk more today, is glaucoma. And the polymorphism of optineurin, M98K, is basically, it sensitizes cells to various kinds of stresses. So I would like to share the journey that how did I reach to this, to make the statement that M98K polymorphism is sensitizing cells to the stress conditions. So let's see what is glaucoma. Glaucoma is a heterogeneous group of optic neuropathies characterized by cupping of optic disc, degeneration of optic nerve, death of retinal ganglion cells, which leads to irreversible blindness. So there are a couple of technical terms. How do physicians identify that there is a glaucoma? So the area of retina through which blood vessels enter into the eye pocket, and the nerves of photoreceptors, to be precisely, retinal ganglion cells, exit the retina. That area, that junction is called as optic cup. And this optic cup is a blind spot inside the retina, and the area which is around that blind spot is called as optic disc. So whenever we say that there is a glaucoma, that means often it is because of the cupping of optic disc. Basically, this is the inflammation caused because of the death of the cells. So when there is a more cupping of optic disc, that means there is a more severe symptoms of the disease. Another hallmark feature of glaucoma is loss of vision from periphery towards the center. That means there is a vision loss from periphery towards the center. That is called as tunnel vision, which is represented here. So glaucoma, the cases of glaucoma are increasing, especially in South Asia, particularly coming from China and India. So this has a huge economic burden on developing countries, particularly like India. So in order to understand and mitigate this economic burden, we need to understand how pathogenesis of glaucoma occurs. Glaucoma is caused by several environmental as well as genetic factors. There are 6 well-defined, well-studied loci which have been shown to be associated with glaucoma, and optineurin is one of them. So glaucoma is of several types. the main classification of glaucoma is primary glaucoma, is high ocular pressure glaucoma, and normal tension glaucoma. So our eye has a three-dimensional shape because it has an intraocular pressure. So when the intraocular pressure increases, that is termed as high tension glaucoma. And when the intraocular pressure is physiological, is normal, then it is called as normal tension glaucoma. And the mutations in optineurin are associated with normal tension glaucoma. So let's see, what is optineurin? Optineurin is basically an adapter protein. It is optic neuropathy inducing protein. It is 577 amino acid protein. It is highly conserved in mammals and expressed in almost all the tissues at differential levels. Mutations in optineurin are associated with several neurodegenerative disorders like familial glaucoma, amyotrophic lateral sclerosis, and more recently, Huntington's disease and Parkinson's disease as well. The molecule optineurin got into more prominence when it has been shown that it is an autophagy receptor. So what is an autophagy phenomenon? So inside the cell, there are two types of degradative systems. One is called as 26S, proteasome-mediated degradation, where E3 ligase ubiquitin conjugation system, which is carried out by E1, E2, and E3, which eventually ubiquitinates the target protein, which gets degraded by proteasome machinery. The other pathway of protein degradation is via autophagy, in which a protein or an organelle or anything which is bigger, which cannot conventionally go through the proteasome system, is directly dumped into the lysosome. So this phenomenon is called as autophagy. And there are very selective proteins which basically carry these organelle or protein or anything which is to be degraded to lysosome. These proteins are called as adapter proteins, in particular autophagy receptors. So optineurin is an autophagy receptor. So how does optineurin do these functions? So optineurin is basically an adapter protein. So it has a coil-coil domain. So most of the coil-coil proteins, they function as an adapter protein. That means they bind or they recruit several proteins at a site of action by serving as a platform. So optineurin does not have any enzymatic activity. It provides a platform for certain proteins to come and interact with each other. And we know that any reaction is nothing but an intramolecular interaction. So that's how optineurin facilitates several reactions. So since optineurin is facilitating several reactions, it is expected that if there are mutations in optineurin, that means the mutant protein may not bind to its interacting partners that efficiently compared to the wild-type optineurin protein, right? So here is a list of several optineurin mutations as well as polymorphisms, which are shown to be associated with glaucoma or ALS. And most of my work revolves around the polymorphism, that is M98K. So as I've mentioned earlier, that in glaucoma, there is a death of retinal ganglion cells. So let's see what are retinal ganglion cells, because these are the cells which are the site of pathogenesis in glaucoma. So in case of development of eye, that is here in case of development of retina, retinal precursor cells gives rise to these 6 different types of cells and these 6 different types of cells are arranged in this fashion inside the retina. The outermost cells which sense the light are basically the photoreceptors, and they are of 2 types, cones and rods. And they absorb the information and then that transmitted to interneurons, which are nothing but basically horizontal cells, amacrine cells, and bipolar cells. And these interneurons eventually transfer the information to retinal ganglion cells, which are the innermost cells in the retina. So the function of these retinal ganglion cells is to transmit the visual information received by photoreceptor to the visual cortex in the brain. So if we want to study specifically retinal ganglion cells, so then we need to understand how to identify retinal ganglion cells. So in retina, there are several proteins whose expression is restricted to retinal ganglion cells. And these are termed as RGC specific markers in retina. So whenever I say RGCs, that means I'm talking about retinal ganglion cells. So these are the different receptors of retinal ganglion cells, which are expressed in retina. So this was a time when I decided to join Dr. Ghanshyam Swarup's lab to pursue my PhD. So one of the challenges at that time was having a right cell culture model. So previously, for retinal ganglion cells, RGC5 cell line has been used more often, and it was supposed to be a rat retinal ganglion cell line. But eventually, several groups have shown that they do not express certain markers of retinal ganglion cells, and that is why the research using RGC5 cells has been in question for a long period of time. And that's where I started pursuing my PhD. Then I decided that the first objective of my PhD should be to identify a cell culture model where optineurin mutations can show a phenotype. Here in this case, phenotype means the functional defects because of these optineurin mutations. So these were my objectives, that is characterization of retinal cell line model to study the functional defects caused by glaucoma-associated mutants of optineurin. Second, that is establishment of human primary retinal cells as a model to study glaucoma pathogenesis. This is important because so far there isn't a single human retinal ganglion cell model that is available. And third one is how optineurin mutations, particularly polymorphism M98K, induces neurodegeneration. But in case of cell culture setup, we always often refer it as cell death. So with these objectives, I have divided my presentation into 3 different subsections. So we'll see one by one. So now coming to the recharacterization of cell line. So I wanted to recharacterize 661W cells because when the labs have immortalized RGC5 cells, they were also culturing 661W cells. And often it has been shown that 661W cells have certain neuronal differentiation potential, and those that basically means that these are a neuroblastoma kind of cells. Here in particular, it is retinoblastoma cells. So, and 661W cells are from mouse retina. So based on these information, I decided to give it a shot that let's see whether 661W cells, whether they have properties of RGCs or not. And if they have RGC specific properties, then what is the consequence of overexpression of these glaucoma-associated mutants of optineurin in them? So as I've mentioned, that there are some proteins whose expression is restricted to retinal ganglion cells in retina. So to begin with, I analyzed both these cell types for the expression of these RGC specific markers. And interestingly, I found that both the RGC5 cells, what we have in our lab, as well as the 661W cells, what we bought, both of them expressed almost all the RGC specific markers. So this is a Western blot showing expression of RNA binding protein with multiple splicing, BRN3 family proteins, NUN, one of the most argued expression protein in the field of RGCs is Thy1 and beta-3-tubulin. We further confirm the expression of these proteins by immunofluorescence. Here we can see that this is not basically any heterogeneous population of two different kind of cells, because all the cells are almost uniformly expressing these markers. So we understood that, well, both these cell types specifically express RGC-specific markers. So then we wanted to, since 661W cells has previously been speculated that they can get differentiated. So here what I did was I analyzed these cells for the expression of RGC precursor cell specific markers. So that means if these cells have the precursor cell specific markers, that means they can have an ability to undergo neuronal differentiation. So these are certain markers of retinal RGC precursor cells, and we found that almost all of them are showing the expression of these markers. Here is the Western blot, and we can interestingly see that there is no expression of this RGC precursor cell specific marker nestin, because these retina is derived from an adult mice. Okay, so we understood that these cells have the expression of RGC precursor cell specific markers. So we potentiated these cells for neuronal differentiation by treating them with staurosporine. Staurosporine is known to induce neuronal differentiation. So if the cells are neuroblastoma and you treat them with the staurosporine, you can see a mature neuron-like morphology. So here you can see that upon staurosporine treatment, the morphology of the cells changes, and we can see the very long neurite-like structures. So we also confirmed what is the consequence of this neuronal differentiation on the expression of RGC-specific markers, and we observed that this neuronal differentiation is indeed increasing the level of RGC-specific markers, suggesting that these RGC precursor cells now are getting mature, getting differentiated into more functionally unique set of retinal ganglion-like cells. So far, we understood that these cells express RGC-specific markers as well as RGC precursor cell-specific markers, and they undergo neuronal differentiation. So then we wanted to check what is the effect of overexpression of glaucoma-associated mutants of optineurin in these cells, because ultimately we want to check what is the functional defect caused by these optineurin mutations in these cells. So we carried out cell death assay. We used it as a readout to understand the functional defects, probably caused because of the optineurin mutations. So the assay we have used here is annexin 5 and 7 AAD staining. So here is the mechanism, how it happens. So in case of normal cells, we have phosphatidylserine, which is on the inner leaflet of the lipid bilayer, and upon apoptotic stimuli, it comes onto the outer leaflet. And then this phosphatidylserine has an affinity towards annexin 5, and this annexin 5 is bound to a dye. So here we can see that if we overexpress a protein, and this protein has here, we can see that two different kinds of fluorescence. One is golden yellow, that is basically annexin 5 staining, and then there is a red, dark red staining, that is 7-AD staining. So these two different type of stainings can be seen depending on at what stages of apoptosis these cells are at. So utilizing this mechanism, we carried out a cell death experiment. So we overexpressed wild-type optineurin and two glaucoma-associated mutants of optineurin, M98K and E50K, and one of the ALS-associated mutant of optineurin. As I have mentioned earlier, that optineurin not only causes glaucoma, but also it is associated with amyotrophic lateral sclerosis. And after overexpression, we stain these cells with annexin 5 and 7 AAD, and we basically counted under microscope manually that what is the percentage of cells which are positive for GFP as well as positive for annexin 5. And this is what we got. So both these glaucoma-associated mutants induce significantly higher cell death in 661W cells compared to wild-type optineurin expressing cells. So since I've mentioned that optineurin mutations are also associated with neurodegenerative disorder, that is ALS. So we carried out similar set of experiment in a neuroblastoma cell line whose origin is from the neuroblastocytes. So this is NSC34 cells are often used for ALS related research. So we overexpress wild-type optineurin and both glaucoma-associated mutants of optineurin, as well as E478G. And in these cells, we observe that only ALS-associated mutant has shown significantly higher cell death compared to wild-type optineurin. So set of these experiments suggest that glaucoma-associated mutants of optineurin are inducing cell death only in 661W cells, but not in NSC34 cells. So basically, this set of experiments gave us a model to work with. So to summarize this part, 661W cells express RGC-specific as well as RGC precursor cell-specific markers. They undergo neuronal differentiation. And both glaucoma-associated mutants of optineurin induce significantly higher cell death in 661W cells compared to wild-type optineurin. So now we had a cell culture model, which is like a continuous supply of cells. But again, the question is, these cells are of mouse origin, and we wanted to have cells which can be of human origin. So while pursuing that, I wrote a review arguing about the properties of 661W cells, that they express markers of certain retinal ganglion cells, certain markers of retinal progenitor cells, and we concluded that 661W cells, since they are expressing multiple markers of precursor as well as RGC-specific markers, these cells can be called as retinal ganglion-like cells and can be utilized to study mechanisms of neurodegeneration and cytoprotection relevant for glaucoma pathogenesis. So then the next objective was to establish human primary retinal cells as a model to study glaucoma pathogenesis. So far, there are no human primary retinal cells available, and the protocols are really extremely tedious, and the yield, what we get from a retina, is very, very less. So in order to get access to human primary retina tissue, we collaborated with Dr. Inderjeet at LV Prasad Eye Research Institute. So in eye research hospital, there are several conditions in which physicians advise the eyes to be removed to minimize the further damage. So we got access to tissues which are from aborted fetuses and from the cadaveric source, or we also got access to some samples which are specifically from anterior staphyloma. So what is anterior staphyloma? Is here we can see that this sclera part of the eye, it has an outgrowth. Basically, the cells which are sclera, within the sclera layer, they start dividing, and that covers the entire eye. So we decided, and often these patients are told to get their eyeballs removed because this can eventually damage the optic nerve and eventually the brain. So we got access to these tissues with the help of Dr. Inderjeet, and we hypothesized that maybe we can culture some of the retinal cells from these eyeballs. So this outer side, which is represented here, is the outer side of the eye, whereas the retina resides as an innermost layer in the eye. And in all the cases of anterior staphyloma so far, there hasn't been a report which mentions that there is any problem in retina. So we hypothesize that maybe from this retina, we can culture human primary retinal cells, and we can check if these cells are indeed human primary retinal cells. Then we should see the phenotype, what we observed in mouse retinal cells, to be able to induce cell death in these cells as well. So we followed this. This is a simplistic view of the protocol with like a couple of steps mentioned here. Basically, you get the eyeball, then you remove retina, which is like more tedious part to get pure retina, and then you basically do a fine chopping of it, and then eventually somehow we grow it. So what we decided was, like, since these cells are primary cells, we knew that they are not going to grow for really long period of time. So we wanted to analyze them at each passage. So here we analyze these cells for each passage and check them for the expression of beta-3 tubulin and GFAP. So here beta-3 tubulin is a retinal ganglion cell specific marker, and GFAP is a Muller glial cell specific marker. So in retina, there is a subset of glia which have been often shown that these are the cells which prevalently survive if you culture the primary cells. So here we can see that in passage 1, we have less number of cells which are positive for the staining of beta-3 tubulin, and more cells which are positive for GFAP. And as we have seen with increase in passage, we saw substantial reduction in the cells which are positive for the expression of GFAP, suggesting that these cells which are eventually left in culture are nothing but the neuronal cells which are surviving from the human primary retinal cells. So what we did is we further analyze. Now the assays are carried out specifically from passage 3 onwards. So basically these cells do not grow after 6 to 7 passage. So we did all of our experiments in that short window of time. So we observed that these cells express BRN family proteins. They are also positive for the expression of other RGC specific markers like RNA binding protein with multiple splicing, NUN, and beta-3 tubulin. So then passage-wise, we analyze the expression of these RGC specific markers and wanted to see what is happening at each passage. So here we can see that these cells are nicely positive for the expression of beta-3 tubulin and all other RGC specific markers. But with increase in passage, we can see that the expression of these RGC specific marker has started going down. So since I've mentioned that we also check these cells for the expression of other retinal cell specific markers, like for Muller glia, we have used GFAP, and for precursor cells, we have used CHX10. CHX10 is also a retinal bipolar cell specific marker. So these cells were completely devoid of expression of these proteins, suggesting that eventually human primary retinal cells surviving in our culture are nothing but RGC-like cells. We also analyze these cells for the expression of RGC precursor cell specific markers. And here we can see that with increase in passage, we see the reduction in the expression of precursor cell specific markers. So these observations suggest that during this short window of passage 3 to passage 6 or passage 5, sometimes these cells express the markers of retinal ganglion cells as well as markers of retinal ganglion precursors as well. So then we wanted to analyze what is the effect of these optineurin mutations when we overexpress them in these human primary retinal cells. So we carried out a similar assay, which I've mentioned before, and counted the cells which are positive for annexin 5 and 7A staining. So here also we can see that the percentage of cells showing cell death are much higher in M98K as well as E50K overexpressing cells compared to wild type optineurin, suggesting that this is a established set of steps and the protocol during which we can utilize these cells to study the functional defects caused by optineurin mutations. So far, this was the first report where we could culture human primary retinal cells and show that these are positive for the expression of these markers, and we can eventually utilize this protocol to generate human primary retinal cells. And this is a very robust protocol. So to summarize this part, during these set of experiments, we observed that human primary retinal cells express RGC-specific markers under defined growth conditions. They express RGC precursor cell-specific markers, and they are sensitive to overexpression of glaucoma-associated mutants of optineurin. So now, coming to the third part of the presentation, that is understanding the role of optineurin and its polymorphism M98K. How does this polymorphism induce cell death in human primary retinal cells or in 661W cells? So before getting into the mechanistic details, I would like to introduce M98K. So what is M98K? Is a polymorphism of optineurin, and it is associated. The most interesting point with M98K is that it is associated with glaucoma only in Asian populations, but not in Caucasian population. So we don't know why. And in some Asian population, in some countries, the occurrence of M98K is as high as 20%, whereas in India, it is still considered as a mutation. So we hypothesized maybe this differential association of glaucoma could be due to the environmental or other genetic factors which are cooperating to cause the death of retinal ganglion cells. So to address that, we have generated 661W cells, which are stably expressing human optineurin, because that's the only relevant cell type which we could figure out. And we generated stable cells by lentivirus-mediated overexpression, because initially I have told you that whenever we overexpress M98K, there is significant cell death. So that was lipofaction-induced overexpression. And in this case, we have generated lentivirus-mediated expression. So in case of lentivirus-mediated expression, the expression starts very slowly compared to the other ways of overexpression. So other than this difference compared to the lipofaction, we check whether these cells express human optineurin because human optineurin is tagged with a flag tag. So here is a Western blot showing that the parental 661W cells, they do not have any flag expression, whereas all the clones have significant expression of optineurin. And optineurin has previously been also shown that it plays a role in cell cycle, cell division, particularly, not cell cycle. So we analyze these clones for their proliferation rate. And we observe that all these cells, all these clones have similar proliferation rate, which is a good thing, because whatever the phenotype we will observe is not because of the differential proliferation rate. So one of the genetic factors which has been shown to be associated with glaucoma, other than optineurin, is TNF-alpha promoter polymorphism. So one of the interesting observations from Japanese cohort is there is a polymorphism of TNF, that is 836A, and this polymorphism, whenever it is seen with M98K, the symptoms are very severe. So we hypothesized maybe this is how optineurin is making cells susceptible to TNF exposure. That means basically it is causing functional defects. But interestingly, in case of glaucoma or in the normal physiological conditions, retinal ganglion cells do not express TNF alpha. The cells which express TNF alpha are the Muller glial cells. So we hypothesized maybe M98K is sensitizing retinal cells to the TNF alpha in case of physiological condition coming from a Muller glial cells. So to test this hypothesis, we treated these clones and analyzed them for their cell survival. So here we have performed two different assays to analyze cell survival. First is MTT assay, which is basically a calorimetric based assay where we count basically number of surviving cells. So the viable cells have this mitochondrial reductase, which convert this MTT to a purple colored compound. And then we can measure the amount of purple color produced by a spectrophotometry. So here we can say that the number of cells surviving upon TNF alpha treatment are significantly higher compared to the mutant ones. So we further confirm this reduction in M98K expressing clones is because of the activation, is because of the apoptosis. So we know that apoptosis, So we know that apoptosis occurs by activation of caspase 3, which is an executionary caspase. So in case of both intrinsic as well as extrinsic pathways, the executionary caspase is caspase 3. So we analyze for the activation of caspase 3 as well as caspase 8. And here we can see that wild type cells treated with TNF alpha, they have a little amount of cell death compared to the M98K clones, where we can see significantly higher cell death. So then we wanted to understand how TNF is inducing cell death. Because TNF alpha can activate two different completely distinct complexes. One of them goes for NF-kappa B activation, which requires active protein synthesis because NF-kappa B is a transcription factor. It activates several cell survival genes. The other complex it activates is complex 2 that induces basically the cell death. So in order to differentiate these two, often people use cyclohexamide. Cyclohexamide is nothing but a protein synthesis inhibitor. So if NF-kappa B is getting activated, that means it has to synthesize proteins, right? So if you treat the cells with TNF, at the same time add cyclohexamide, then you are literally blocking NF-kappa B pathway. So this rules out the possibility that there is no involvement of NF-kappa B. So what we did here is we chose one clone and treated them with a different time points with cyclohexamide and TNF and plotted it for caspase 3 as well as caspase 8 activation. And here we can see that from 4 hours itself, we have substantial cell death in M98K clones compared to the wild type clones. So these observations suggest that M98K is sensitizing cells to TNF alpha, making them more susceptible to the TNF alpha exposure. The other stress condition which we wanted to explore was ER stress. ER stress is nothing but an imbalance of protein synthesis and protein folding, which leads to accumulation of various misfolded proteins within the ER. So often ER stress has been studied extensively in case of high tension glaucoma. So far, there was only one observation where it has been speculated that maybe normal tension, in case of normal tension glaucoma, also ER stress might be a contributing factor. So in this set of experiments, we tested the involvement of ER stress in normal tension glaucoma. So what happens when there is ER stress? So let's say there are tons of proteins which are misfolded. So this ER stress caused because of the misfolded proteins is sensed by a sensor that is called as GRP78. And this master regulator of ER stress in normal physiological condition keeps these sensors of ER stress, that is PERK, ATF6, and IRE1 alpha inactive. So whenever there is ER stress, whenever there are misfolded proteins which are accumulated in the ER, GRP78 binds to them. Since this GRP78 is now bound to these misfolded proteins, there is activation of these sensors of ER stress, that is PERK, ATF6, and IRE1 alpha. So all these three sensors have different signaling events which activate different sets of proteins which can help to cope up with ER stress. So artificially, in cell culture condition, we can create a situation which mimics ER stress is by treating the cells with tunicamycin. So basically what tunicamycin does is it inhibits the formation of phosphodiester bonds between two. So basically it inhibits the formation of a folding of a protein by inhibiting certain reductases in the ER. So we treated these clones of M98K and wild type with ER stress inducer, that is tunicamycin, and the MTT assay shows that M98K expressing cells have bare minimum survival rate compared to the wild type expressing cells. We further confirm this cell survival assay by analyzing them for the activation of caspase 8 and caspase 3. So this is the quantification of caspase 8 and caspase 3, where we can see that compared to wild type, the activation of caspase 3 and caspase 8 is more than 3 to 4 fold. So this is a dramatic cell death caused by cell death observed by M98K expressing cells. So then we wanted to see what is the impact of this ER stress, basically how and why M98K cells are more susceptible to ER stress. So we analyzed the levels of ER stress sensors and signal transducers. One of the bona fide marker of ER stress is CHOP. So here we can see that upon tunicamycin treatment compared to the wild type expressing cells, there is 4-fold higher induction of CHOP. And we did not find any change in the levels of GRP78. But most interestingly, we found that the levels of ER stress sensor IRE1 is almost twice as compared to the wild type expressing cells. I'm not showing all the data here, but we also have the data for higher levels of PERK as well as ATF6. So these are the quantifications from set of 3 different experiments, and you can see that majority of them, all of them, show significantly higher induction of all these ER stress sensors specifically in M98K inducing cells, M98K expressing cells. So then we wanted to analyze a lot of other genes. So what is the impact on them? So the easiest way to do that was to do a qPCR. So here we can see that a similar experiment was performed, but this time we analyzed the level of the transcript, the level of mRNA at different time points. So certain proteins, we interestingly observe that they are higher even at a basal level itself. So one of them is IRE1 alpha, PERK, and XBP1. So other than these 3 proteins, we also observe that there are some more ER stress response proteins which are also differentially expressed in M98K expressing cells. So as I've mentioned in this figure, that all these three different sensors, let's say if all the three of them are differentially being regulated, then we should be able to see all these downstream signaling events differentially in wild type compared to M98K expressing cells. So in order to analyze that, we have carried out different assays. So under the sensor IR1 alpha, there is a splicing of XBP1. XBP1 is a transcription factor, which upon ER stress gets spliced. Basically, there is a removal of 21 nucleotides, and then it gets into the nucleus, and it basically activates sets of genes which are like ER stress chaperones and other proteins. So here we treated the clones of wild type NM98K and isolated RNA from there and analyze and run an agarose gel after amplifying the transcript. So this set of primers amplify both the transcripts, the spliced one as well as unspliced one. So the spliced one is a smaller, so it runs faster. So here we can specifically see in case of M98K post 24 hours of tunicamycin treatment, this band is more brighter compared to the counterpart of wild type optineurin expressing cells. So this suggests that XBP1 splicing is also more in case of M98K expressing cells. Similarly, the other sensor, that is ATF6, we analyze its activation as well. So how does ATF6 gets activated? It gets activated upon ER stress by visiting Golgi. So in Golgi, it gets cleaved. So the actual molecular weight of ATF6 is somewhere around 100 to 100 and I think it is 98, but it gets glycosylated, so it runs higher than its molecular weight. But during the conditions of ER stress, this gets cleaved to 57 kD fragment. So here we can see that only in M98K expressing cells, there is cleavage of ATF6, suggesting there is more activation of ATF6 in M98K expressing cells. So then we wanted to understand why these sensors are higher in M98K expressing cells, why this signaling is more in M98K expressing cells. So in order to understand that, we hypothesized maybe there is a role of autophagy in regulation of these sensors. So as I've mentioned that optineurin is an autophagy receptor. And in case of M98K, previously our lab has shown that optineurin polymorphism, M98K has higher autophagy activity. So how do we measure autophagy? Is by analyzing the relative ratio of LC31 to LC32. So what is LC3 molecule? So when there is induction of autophagy, this LC3 molecule gets lipidated, and this lipidated molecule runs faster on a SDS page. So we treated these cells with tunicamycin, and here is the plot for LC3. We can see that compared to the wild type optineurin expressing cells, the mutant expressing cells have higher level of LC32. This suggests that the wild type expressing cells have more autophagy. So then we wanted to further confirm this is the mechanism through which the levels of ER stress sensors are being regulated in our experimental conditions. We carried out ATG5 knockdown assays. So in brief, you can remember that every autophagy that occurs, in 90%, it depends on ATG5. It's like a core complex of autophagy. But there are some autophagies which are ATG5 independent as well. But we know that optineurin mediated autophagy is so far ATG5 dependent. So as I've mentioned that we observed that higher level of sensors of ER stress in M98K expressing cells compared to wild type expressing cells. So we did the knockdown of ATG5. Basically, it was siRNA mediated knockdown of ATG5. And we can see that the level of PERK as well as IRE1 are reduced upon knocking down of ATG5. So this is literally a counter regulation which we observe compared to the whatever has been proposed before. So this experiment suggests that M98K cells have more autophagy, and as a consequence of that, we have more levels of ER stress sensors. So these observations, based on these observations, we propose this model that whenever there is ER stress in M98K expressing cells, they have higher levels of activation of ER stress sensors as well as ER stress transducers, which eventually causes a high activation of caspase-3 as well as caspase-8, causing cell death. So based on these observations or ingest of my PhD, that we observed that 661W cells express RGC-specific markers, and they can be utilized as a cell culture model to study mechanisms of neurodegeneration and cytoprotection relevant for glaucoma pathogenesis. And we could establish a protocol to culture human primary retinal cells. And towards the end of my PhD, I could show that M98K cells are sensitized due to ER stress and TNF-alpha, which might be contributing to the differential association of M98K to glaucoma. So whatever studies or independent parts of my studies are published, they are listed here. And with this, I acknowledge my supervisor, Dr. Ghanshyam Swaroop, our collaborator, Dr. Indrajit Kaur, my co-supervisor, Dr. Nishant Jain, and some of my other colleagues who have helped me in this work, and my current mentor, Dr. Michaela Gack. With this, I would be happy to take questions from you. Thank you.
Okay, thank you, sir, for very nice and explanatory lecture on your own topic. I request all the participants to ask your queries to Dr. Jubair, sir. Yes. One more request to Dr. Jubair, sir. Please share the knowledge regarding opportunities of these research students, because they are just joined to their PhDs. So what are the opportunities, particularly in foreign? If you know, please share with our students, sir.
Oh, well, in terms of opportunities, you need to decide now. These days, doing postdoc is not that trivial. So people often go to industries, especially in the West or even in US. People don't stay in academia after PhD. They just go to industries. Now, there are so many opportunities in different stream of the work, like science communication, or you can go to industries if you don't want to continue or want to have a lab at the end of the day. So the opportunities are enormous, especially with increasing the big data analysis kind of thing. So the people who do that kind of study, or they want to pursue bioinformatics, they now with a lot of virtual platforms, we can study bioinformatics, and then people are able to start their own startups as well. So there are lots of opportunities, but we need to decide what do you want to do ahead of time, so that you can pursue or you can understand those streams in the beginning itself. Like, as I've mentioned, that when I was doing my BSc, I knew that after probably when I was in the vacation, summer vacation of BSc second year, I had a bit of understanding that, okay, fine, I will do a master's and then eventually I will do a PhD. but I didn't know that how am I going to reach there. Then when I had a target, then I started exploring about how I can reach to that stage. So it is important for you to understand what kind of life you are expecting from yourself, what kind of science you are expecting from yourself, and what are your levels of commitment towards your career. So these are important things. If you have those things figured out, then you will find ways how to reach there. And there are so many people which are available on the professional networking groups who are happy to help you, who are happy to share how to reach there. So it is important first to have a clear understanding that what you want to do with your PhD or towards the end of your PhD. So if anybody has any questions about how to, if they want to stay in academics, what are the requirements? If they want to go to industry, what are the requirements? Then we can discuss about it separately.
Okay, thank you, sir. Dear participants, please feel free to ask your difficulties, because it is very important to know what are the difficulties faced and all these parameters, because Dr. Jubair is our alumni, and we are really proud of you, sir. And really, it is much more beneficial for our students that if you guide them regarding their PhD and all these things. So it is very, very important. So dear participants, please ask. If you have any queries, either you may ask in, I think you may know Hindi.
Yeah, that's perfectly fine. I'm fluent with Hindi, Urdu, Marathi.
Yeah. So dear
I'm okay with
participants,
it.
please ask in Hindi also. No problem. विचारा काही प्रश्न असतील? Yes.
Okay, if you are not asking, I will share something. So
Yeah.
very often in academics, it is it is kind of frustrating if you don't get good results, and especially if you don't get good results in like first maybe couple of years, or maybe third year or fourth year, because all these every year, all these research institutes, and probably, I don't know the system now, but they will be an open presentation. So if you don't have result, then you are standing in front of 70 people, and then they are going to ask, oh, that's what you did in 1 year. But unfortunately, things don't work, and it is really, really embarrassing situation. So when I was working on this project, one of the main objectives which we excluded eventually was like generation of a transgenic mice with M98K and having all these similar objectives and look it into a physiologically relevant system. So for 3.5 years, I used to go to Animal House almost every day, used to do a genotyping almost every other week. And after 3.5 years, maybe I would have screened almost 500 mice, close to, and we ended up with not a single M98K transgenic. We don't know what is the reason. So that was the time when we decided that probably I need to think about what I want to do in my PhD, because the things were not going the way we planned. So it can be frustrating a lot at times. But then that was the time when I decided that I should focus more on the cell culture work. And then eventually, within probably, I would say within a year or so, I had my first publication. And then that actually boosts your enthusiasm towards pursuing your work. And then the second incident was like the COVID-19 pandemic, right? We all in CCMB volunteered for COVID-19 screening because we did not have access to the labs. So for almost maybe 12 months or so, we literally did not do anything in wet lab, just because there was one paper which was like in communication. So I think on entire wing, I was the only one who was fortunate enough to get a permission from the director to work. But that period has literally increased my PhD from 5 years to 6.5 to 7 years. So these are the kind of things which you need to be ready to face and still say that, okay, fine, I want to still continue working in academics. So this is something which you should be prepared if you really want to be in academics. I'm not scaring you, but few people find it very smooth, but few people don't. So if you don't find things going that smooth, then you need to be patient enough to take it.
Thank you, Dr. Jubair. Again, any questions, queries? You may ask your queries in chat box also. No problem.
Yeah, that would be nice.
Yes. Because such opportunities not comes again and again. Dr. Jubair has spared his valuable time for us, as I think it is midnight at America. Yes,
You
USA?
should, yes, you should ask me because it is almost 3 o'clock here. So make use of the time.
Oh,
Yes.
yeah. Okay. One question in chat box, that how you
Okay.
approach for pursuing postdoc in USA.
Okay, so how do we approach for postdoc in US? So it's not specific to US. You can approach in a similar way to Europe as well as anywhere else. So often we go to conference. As I went to the conference in Canada for presenting some of my work, I met a couple of people over there who came to my poster, and then I discussed with them. And then I also mentioned that, oh, well, this is the thing and blah, blah, blah. And I'm towards the end of my PhD, and I'm looking for the postdoc opportunities. And then a couple of them have responded me. But then that is one way how you find or how you interact with the potential mentors in a conference. The second one is like when you are like close to writing your thesis, or when you are done with your thesis, because it takes a while for you to have your viva done. So that's the best time for you to start writing to the labs in which you are interested in. Write them a nice piece of email where you will describe a bit of yourself, your work, and most importantly, if your email is without the interest of the lab, they don't even bother to respond to you. So if your email has something which is significantly associated with their lab or what they are doing, that's the time when they will start responding to you. Otherwise, they will not respond. So these are the two ways in which I happen to get an opportunity to reach here.
So thank you, Dr. Jubair sir, for sparing very valuable time to our students in spite of midnight at your place. Still, you are with us for sharing your knowledge and just giving a lecture just because of the love and affection with our college. Thank you very much, sir.
Sure.
On behalf of Dr. Borkar madam, especially, I express my gratitude to you, sir, as well as on behalf of all the participants and research committee, Science College Nanded. I must express my gratitude to you, sir, for sparing your knowledge, for sparing your valuable time for our research students. Whatever the things you have teach to them, they must very much important for their research work. They must follow all the tips you have given in their research work. So I must thank to Dr. Jubair sir, and in future also we will call you, sir, not online, but we would like to call you offline. If whenever you visited to India, we will.
So every year I visit India, but unfortunately this year I think I came to India in February. I couldn't see Borkar Ma'am because I met an accident. I couldn't travel. I was not in a position to walk. So that's why I didn't visit the Science College. Otherwise, I would visit Science College all the time.
Yeah, actually, Borkar Madam is in traveling, so that's
Yeah,
why
I know. She texted me.
She is not able to give vote of thanks. That's why she asked me. You mentioned.
Sure.
Thanks. So thank you, sir. Thank you.
I think I can see a question here.
Yeah.
Social media handle. Okay. You can search my name in LinkedIn. You can find it easily. Yeah.
Okay.
You can search my name in LinkedIn. You will get it. Yeah. Search with CCMB as a tagline. You will find it. Yeah.
Okay. I will share your resume. I have resume of Dr. Jubair sir. I will share with all the students so that they will search all the information regarding you.
Yeah, they can do that. Yes.
Yeah. So thank you, sir. We are not taking your much more time.
That's all right.
It is midnight at USA. So thank you very much, sir.
Yeah. Thank you for giving me the opportunity. Yeah.
Okay. Thank you, sir. Thank you.
Bye-bye. Good luck.
Okay. So dear participants, now it's time to move towards our next session. 1 minute, Dr. Gingine sir will join very shortly. So Dr. Gingine sir will join very shortly. If you have any difficulties, please ask till then. Yes. Hello. 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 Gingine sir. Sir. Ganesh. Ganesh, please make co-host to Dr. Gingine sir. Sir, Gingine, sir.
Yes,
Okay.
yes.
Okay, sir. So, dear participants, today we have with us Dr. Ashok Gingine, sir, Assistant Professor in School of Educational Science. I think we know him because he has given all the entire notes and all these question papers to you. So I welcome Dr. Gingine, sir, on behalf of all the participants and on behalf of Research Committee Science College Nanded. And I request Dr. Gingine, sir, to start his presentation. Sir.
Yes. My screen is visible now. Anyone tell me?
Yes, sir. Yes.
Okay. Thank you, sir. Good afternoon, all of you. Today we discuss regarding important questions and research tools, qualitative research tools and quantitative research tools. As per as the concern of first, which was the important area, which was the important aspects regarding examination views. These are the format. These are the structure of question paper. Question number first is compulsory short notes. This type of short notes ask solve any 4 short notes from remaining 5. Means previous so many research paper frequently repeated questions that such like secondary source of data, what is the types of data, what is sources of data, primary source of data, secondary source of data, nature of data, means qualitative type of data, quantitative type of data, as per the concern of that type of short notes asked in previous question. Case studies, types of case studies, characteristics of case studies, observation, characteristics of observation, types of observation, this type of short questions asked in previous question. These are the important aspects in future. Maybe last week of June held on coursework examination, that is the tentative date. Types of population means homogeneous population, heterogeneous population. These are the asked on short notes. Definition of research and characteristics of research, research steps, scientific research steps, and references and bibliography. These are the important aspects as per the concern of question number first. And alternative questions asked in future question paper. What is sampling? Explain the types of sampling. What is sampling? What was the difference between probability sampling and non-probability sampling? What is sampling? What was the characteristics of ideal sampling? And discuss the one type of research sampling. म्हणजे सॅम्पलिंग वर एक प्रश्न येणारच आहे किंवा अल्टरनेटिव्ह जर सॅम्पलिंग वर प्रश्न नाहीच आला तर हायपोथेसिस वर आहे. 3 सेट पैकी कुठला तरी एक सेट पडेल. जर ह्याच्यात प्रश्न असेल, हायपोथेसिस वर प्रश्न असेल तर सॅम्पलिंग वर प्रश्न असणार आहे किंवा एका सेट मध्ये दोन्ही प्रश्न येण्याची शक्यता आहे. Isn't it? And next one is, what is a research synopsis? Explain the elements of research synopsis or research proposal. म्हणजे काय, संशोधन आराखडा लेखनाचे घटक स्पष्ट करा किंवा संशोधन आराखडा म्हणजे काय आणि संशोधन आराखडा लेखनाचे घटक स्पष्ट करा, असा लॉंग क्वेश्चन एक येणार आहे. किंवा ह्याला और कुठला आहे, व्हॉट इज रिसर्च रिपोर्ट? एक्सप्लेन द नॉर्म्स अँड स्टँडर्ड्स ऑफ रिसर्च रिपोर्ट. हा जर प्रश्न नाही आला तर ह्याला और प्रश्न कुठला आहे, रिसर्च रिपोर्टचा आहे. जर हे येईल किंवा ते येईल. ठीक आहे. अँड नेक्स्ट वन इज क्वेश्चन नंबर 4. Explain the any one research methods. What is historical research methods? Explain the characteristics of historical research methods and merits and demerits of historical research method. That type of question asked in previous question paper. These are the 3 research methods as per the concern of examination views. That historical research methods. That methods was very important. First one is historical research methods. Second one is the descriptive research methods or survey research methods. What is survey research method? Characteristics of survey research methods, steps of survey research method, types of survey research methods, merits and demerits of survey research method. Any one angle or two angles asked in that questions. And third one is experimental research methods. What is experimental research methods? Discuss the various steps of experimental research method, like types of experimental research methods, systematic steps of experimental research methods, merits and demerits, or merits and demerits of experimental research method or experimental research design. That type of question asked in future question paper. Means these are the three research methods is very important as far as the concern of examination views. ह्या तीन पैकी एक 100% नाही 1000% येणार आहे. हिस्टॉरिकल रिसर्च मेथड असेल, डिस्क्रिप्टिव्ह रिसर्च मेथड असेल, किंवा तिसरी जी आहे ती एक्सपेरिमेंटल रिसर्च मेथड असेल. ठीक आहे. आणि ह्याचे काय पाहिजे? कॅरेक्टरिस्टिक्स, टाइप्स, मेरिट्स, डिमेरिट्स आणि स्टेप्स हे 5 मुद्दे पाहिजे. You deeply studied as the 5 angle of the historical research method, characteristics of historical research method, steps of historical research methods, type of historical research methods, merits and demerits of historical research method, and same way descriptive research method and experimental research methods. And question number 5 or 6, maybe sometime 5 questions asked on numerical data analysis, or sometime 6 question number 6 asking numerical data analysis, isn't it? Means 8 mark weightage or 7 mark weightage divided, or sometime 10 or 5, whatever it may be, as per as the concern of paper setter. म्हणजे काही वेळेस 8 मार्क आणि 7 मार्क अशी फोड करतं, काही वेळेस 10 मार्क आणि 5 मार्क अशी सुद्धा फोड केलेली आहे. डिवायडेशन जे आहे, काही वेळेस 5वा प्रश्न विचारलेला आहे, काही वेळेस 6वा प्रश्न विचारलेला आहे. काही सेटमध्ये 5वा प्रश्न आहे, काही सेटमध्ये 6वा प्रश्न आहे. परंतु 15 मार्काचं वेटेज जे आहे as per the concern of data analysis and testing of research hypothesis, these are the 5 parameters in examination views that is important. First one is a t-test statistical parameters, one-tailed t-test statistical parameter, two-tailed t-test statistical parameters, or testing of null hypothesis through a t-tailed statistical parameters. And second one is a chi-square statistical parameters used in a testing of your research hypothesis, one-tailed t-test, two-tailed t-test, or one row chi-square or double row chi-square. Only observed frequency sometimes reflect in that question, or sometimes expected frequency or observed frequency, or 2-line questions, 2-line chi-square data was reflect in that questions, or mean, mode, median, standard deviation, quartile deviation. These are the important statistically parameters as per the concern of examination views. These are the 5 statistical parameters. Focus on that 5 statistical parameters. And next one is what is research tools? Discuss the qualitative and quantitative research tools. What is research tools? Discuss the any 2 research tools like questionnaires, observation, schedule, checklist. These are the very popular research tools generally used in each and every research college. Means one questions 100% asked on research tools, whatever may be research tools. As per the concern of examination views, these are the 4 or 5 research tools is very important. First one is questionnaires, characteristics of questionnaires, types of questionnaires, merits and demerits of questionnaires, structure of questionnaires. Second one is interviews, definition of interviews, characteristics of interviews, types of interviews, merits and demerits of interviews, and what type of research data you have to collect through an interview. And third one is a checklist or schedule. Checklist, what is checklist? Characteristics of checklist, types of checklist, merits and demerits of checklist. And fourth one is observation. What is observation? What is scientific observation? Which instrument generally used in observation, types of observation, characteristics of observation, merits and demerits of observation. Or sometime what is research tools and difference between qualitative research tools and quantitative research tools. अशा सुद्धा प्रकारचे पूर्वीच्या प्रश्नपत्रिकेमध्ये प्रश्न आलेले आहेत, तुम्ही पाहू शकता. संशोधन साधनं म्हणजे काय? माहितीचे संशोधन साधनं, गुणात्मक संशोधन साधनं व संख्यात्मक संशोधन साधनं यांच्यातील फरक स्पष्ट करा. अशा प्रकारचे प्रश्न हे पाठीमागच्या प्रश्नपत्रिकेमध्ये आलेले आहे. म्हणजे 15 मार्काचं वेटेज हे रिसर्च टूल साठी हमखास प्रश्नपत्रिकेमध्ये असतंच. येस, मीन्स 50 मार्क वेटेज फॉर द रिसर्च टूल्स. अँड नेक्स्ट वन इज क्वेश्चन नंबर 7. What is a research hypothesis? Explain the need and importance and types of research hypothesis. One question asked on a hypothesis. What is hypothesis? Explain the characteristics of hypothesis, types of hypothesis, and testing of your research hypothesis. That type of question asked in a previous question. Means hypothesis, that aspects is very important as per the examination views. Otherwise, explain the types of research. That is an important research area or important concept angle of the that area. Types of research. What is research? Explain the various types of research, like basic research, applied research, action research, or another 3: disciplinary research, multidisciplinary research, or interdisciplinary research. That types of research one questions may be asked on a future question paper. And last one is question number 8. Another short notes, like various types of errors, various like unit number first means meaning, nature, foundation of research, interdisciplinary foundation of research, disciplinary foundation of research, philosophical foundation of research, psychological foundation of research, and sociological foundation of research, scientificness, scientific studies, and meaning of research, nature of research, formulation a research problem, review of research related literatures, objectivity of any concepts, or qualities of good researcher, qualities of good research report, qualities of research sampling methods. That type of short notes asked on a question number 8. म्हणजे हे जे तुम्ही 8 प्रश्न आता जरी पाठ केलेत किंवा अभ्यास केलेत तर तुम्ही 100% पास होणार आहात. फक्त तुम्हाला काय करायचंय, एरिया लक्षात ठेवायचे, कुठले कुठले एरिया महत्वाचे आहेत परीक्षेच्या अनुषंगाने. आणि 91% हे पासिंग रेट आहे, 9% स्टुडंट फेल रेट आहे. बरोबर आहे, म्हणजे एरर आहे की 9% नापास होतात पाठीमागच्या 2 प्रश्नपत्रिकेचं. जर तुम्ही विद्यापीठाच्या वेबसाईटवरनं रिझल्ट काढला आणि त्याचं अनालिसिस केलं, की रिसर्च डेटा म्हणून जर अनालिसिस केलं तर अशा प्रकारचे आहेत. Any difficulty regarding the question structure, format, or area? Yes, anyone tell me? नंतर आपण रिसर्च टूल्स कडे जाणार आहोत. येते आवाज माझा? आकाशजी, आकाश चव्हाण, आहे काय अडचण? यस, आवाज येतो का? विरूपा, आनंदजी, येतोय का आवाज? नाही. एनीवन टेल मी? यस, अनम्यूट करता येत नाही का तुमच्याकडे?
Yes, sir.
Yes.
Audible.
Yes, yes. आहे काही प्रश्न या संदर्भात? चलायचं पुढं. एनी क्वेश्चन रिगार्डिंग दॅट क्वेश्चन सेट और क्वेश्चन एरियाज? In few minutes. Yes, my screen is visible now. दिसते का screen?
Yes, sir.
Okay, thank you. What is research data? What is research data tools? Firstly, we discussed regarding nature of data, types of data, and then we discuss research tools, types of research tools, merits and demerits of research tools. These are the 4 important research tools. First one is questionnaires, checklist, interview, observation, and another supporting research instrument, isn't it? These are the various important rating scales, questionnaires, interview, observation, and checklist. These are the 4 research tools generally used in each and every research process. What is research data? Concept of research data, types of research data, and which norm science standards and which measurement scales using in a research data. That is important. Sometimes some short notes ask on a measurement scales. Which types of measurement scales generally used in a data measuring, isn't it? Means what is concept of research data, isn't it? Data is a individual pieces or information that will be recorded, that will be a measurable, or that will be a not measurable. The purpose of analysis, a raw information from which a statistically area that will be created and that will be a measurable concept. It is called a research data. Sometimes qualitative research data or quantitative research data. Some pieces, some area that was nature of your research data, means qualitative research data or quantitative research data. Whatever may be your information, that information is a measurable information. Measurable information is considered as your research data. Whatever may be, such as measurable or statistically measurable research information, discussion, calculations, or reasoning, basis on a reasoning, that concept is considered as a research data, isn't it? These are the some types of research data generally used in each and every research process. Means first one is a categorical research data, and second one is a numerical research data. Categorical research data generally literature student, linguistic research scholar was use a categorical types of research data. Or basic sciences research student, basic sciences research scholar was use a numerical research data. Means qualitative research data that considered as a categorical types of research data, and quantitative research data that considered as a numerical research data. And these are the 4 measurement scales generally used in each and every measuring a research data. Means categorical type of research data or numerical type of research data, isn't it? म्हणजे जर तुमची माहिती शब्दांमध्ये असेल, बरा, वाईट, चांगला, that is a categorical type of research data. आणि तुमची माहिती जर संख्यात्मक मध्ये असेल, 2, 5, 6, 10 वगैरे वगैरे, that is a numerical data. म्हणजे भाषा विषयातले जे काही संशोधक आहेत ते काय करतात, categorical टाइपचे रिसर्च डेटा ते कलेक्ट करतात. आणि बेसिक सायन्सचे जे स्टुडंट आहेत ते categorical टाइपचे भी करू शकतात आणि न्यूमेरिकल टाइपचे करू शकतात. मीन्स in one research process you have to select a both type of research data, means qualitative research data as well as quantitative research data. Both type of research data you want to use in your research process, isn't it? First one is nominal measurement skills, ordinal measurement skills, interval measurement skills, and ratio measurement skills. These are the 4 measurement skills used in a data measurement process, isn't it? म्हणजे ह्या काय मापन शलाका आहेत. म्हणजे माहितीचं वर्गीकरण किंवा माहिती मापण्यासाठी या शलाक्यांचा वापर केला जातो. जसं आता उंची असेल ती सेंटीमीटर मध्ये मापली जाते, वजन असेल किलोग्राम मध्ये मापलं जातं. तसं माहिती मापण्याच्या ह्या ज्या आहेत त्या 4 पद्धती आहेत. ठीक आहे. फर्स्ट वन इज कॅटेगोरियल रिसर्च डेटा. बरोबर आहे. त्याला काय म्हणायचं, नामांकन शलाका. कॅटेगोरियल रिसर्च डेटा मीन्स व्हॉट एव्हर मे बी वर्ड्स यूज फ्रॉम अ डेटा कलेक्शन्स, व्हॉट एव्हर मे बी terminological words or theoretical words generally used in a categorical type of research data means divided into, yes.
सर स्लाइड चेंज नहीं हो रही है, डेटा कलेक्शन टूल फर्स्ट जो स्लाइड है वही शो हो रही है सर.
ओके ओके, एकच मिनिट आहे. अगोदर सांगायला पाहिजे तुम्ही. ठीक आहे, नाही आता दिसते का? एनीवन टेल मी?
Yes, sir. Yes.
एकच मिनिट. येस, आता दिसतंय? माय स्क्रीन इज विजिबल नाउ?
Yes, sir. Types of data की slide दिख रही है.
यस 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 यस. हाँ, तो ये जो ऑनलाइन टीचिंग लर्निंग के डीमेरिट्स है, जब भी कोई दिक्कत आती है तो आप मुझे बता दीजिएगा. ठीक है. Types of data, these are the two types of data generally used in as per the concern of nature of your research data. माहितीच्या वैशिष्ट्यावरनं हे दोन प्रकार पडलेले आहेत. फर्स्ट वन इज categorical data, second one is numerical data. Categorical data is considered as a quantitative research data or qualitative research data. म्हणजे ही काय, क्वालिटी मेजरमेंट करण्यासाठी हा डेटा वापरला जातो आणि इथं क्वांटिटी मेजरमेंट करण्यासाठी हा डेटा इथं वापरला जात असतो. फर्स्ट वन इज नॉमिनल मेजरमेंट स्केल्स, ऑर्डिनल मेजरमेंट स्केल्स, इंटरवल मेजरमेंट स्केल्स अँड रेशियो मेजरमेंट स्केल्स. These are the some data measurement scales used in a data measurement and analysis of data, tabulation of data, classification as per the concern of these measurement scales used in a data analysis or measurement of data analysis through that 4 types of measurement scales, isn't it? Means कुठली असते ही, गुणात्मक माहिती असेल किंवा संख्यात्मक माहिती असेल, ही माहितीचे दोन प्रकार आहेत. ह्याच्यावर सुद्धा शॉर्ट नोट आलेले आहेत. कॅटेगोरिकल टाइप ऑफ डेटावर सुद्धा शॉर्ट नोट आलेली आहे, न्यूमेरिकल टाइप ऑफ डेटावर सुद्धा शॉर्ट नोट आलेली आहे. किंवा व्हॉट इज रिसर्च टूल्स, एक्सप्लेन द क्वालिटेटिव्ह अँड क्वांटिटेटिव्ह रिसर्च टूल्स. म्हणजे categorical type of research tools and numerical type of research tools, both the type of research tools you have to explain in that question, isn't it? And these are the 4 measurement scales generally used in a research data measurement. Yes, दिसती का आता खान जी?
Yes, sir. Yes, sir.
हाँ हाँ. Categorical research data means whatever may be words or what some themes you have to use as per the concern of that words. You have to collect that data and you have to measure that data. Means whatever may be the gender, language, persons, that type of words you have to use in a measuring a research data. Or sometime means numerical values you have to use in that a research data measuring. Means 01 consider a female, or 0 considered as a male, or 03 considered or 02 considered as a third transgender or third gender, isn't it? That type of words we have to use a measuring a data, that is called a categorical data. Some basic sciences students, you have to use a categorical data, isn't it? Means whatever may be norms you have to use in a measuring a research data. Means third row elements and second row element you have to use in your research process. Suppose केमिस्ट्रीचे मुलं आहेत, थर्ड रो एलिमेंट मधला एक तुम्ही एलिमेंट घेतलेला आहे आणि फर्स्ट रो मधला एक एलिमेंट घेतलेला आहे, व्हॉटएव्हर मे बी दॅट टाइप यू हॅव टू यूज अ कॅटेगोरिकल रिसर्च डेटा. किंवा फिजिक्सचा विद्यार्थी आहे, त्यांनी काय केले की 2-3 वेगवेगळ्या प्रकारचे मॉलिक्यूल घेतलेले आहेत, बरोबर आहे, आणि त्याच्यामध्ये काय केले की ते वर्ड जे आहेत ते तुम्ही वापरलेले आहेत. किंवा आता खान जे आहेत त्या कुठल्या फिशरीच्या आहेत, त्यांनी काय केले 3 वेगवेगळ्या प्रकारचे फिशेस घेतलेले आहेत. यू हॅव टू सिलेक्ट 3 डिफरंट टाइप ऑफ फिश that consider as a categorical type of research data. You want to use in a categorical measurement scale in a using a research data, isn't it? सोपं आहे. म्हणजे काहीतरी शब्दांचा वापर करून तुम्ही माहितीचं विश्लेषण, मापन किंवा वर्गीकरण करणार आहेत. रिसर्चर यूज सम वर्ड्स, सम व्हेरिएबल्स, सम त्याला काय म्हणतात, whatever may be techniques you have to use in a word techniques for measurements of research data, analysis of research data, and check the comparison between one to another. A ratio you have to use a categorical type of research data. That time researcher use a categorical measurement scales. In categorical research measurement scales, first one is a nominal research data or nominal research measurement scales using that particular research data. Means categorical data and nominal data. You have to use both measurement scales in your research process, like a male and female. Means whatever may be Ramesh, Khan, Moshin, Pankaj, Shubhangi, Aisha, that type of words you have to use in a nominal measurement scales. Nanded, Parvani, Aurangabad, isn't it? That is called a nominal measurement scales. Means Swami Ramanand third university research scholars and out of university research scholars means 90% research quota or 10% research quota out of university quota or home university quota, that also use a nominal research data. म्हणजे नावावरनं काय केले आहे त्याचं वर्गीकरण केले, जसं आपण सांगतो की 90% जागा ज्या आहेत त्या होम युनिव्हर्सिटीसाठी आहेत, अदर युनिव्हर्सिटी आहेत आणि अदर दॅन युनिव्हर्सिटीसाठी आहेत, अदर स्टेटसाठी ज्या आहेत. That time researcher use a nominal measurement scales using your research data. बरोबर आहे. कुठली असेल की कुठल्या तरी शब्दांचा किंवा नावाचा वापर करून माहितीचं मापन केलं जातं, जसं बरा, वाईट, चांगला, अति चांगला, अति खराब. That type of measurement scales used in a nominal measurement scales, isn't it? Means satisfactory, unsatisfactory, fully satisfactory, fully unsatisfactory. That type of words we have to use in a nominal measurement scales. कुठली असते म्हणजे काय की qualitative or quantitative, in both type of measurements researcher can use a nominal measurement scales. कुठली असते म्हणजे एका समजा बॉटनीचे विद्यार्थी आहेत, एका फॅमिली मधला एक प्लांट घेतलेला, दुसऱ्या फॅमिली मधला दुसरा प्लांट घेतलेला आणि त्या फॅमिलीचं नाव आणि प्लांटचं नाव तिथं लिहिणं, त्याला काय म्हणायचं, नॉमिनल मेजरमेंट स्केल. Isn't it? Any difficulty regarding particular research measurement scales and nominal measurement scales? Are you understand the concept of both type of measurement scales? Yes, anyone tell me? लक्षात आली का भाग्यश्री जी? गोविंद भोसले सर, शिवकन्या जी, आर यू इन क्लास? दुसरं लेक्चर असलं की कॉमेडी होण्याची शक्यता आहे किंवा फक्त डिव्हाइस तुमचे जॉईन असतील. विरूपाजी, ज्ञानेश्वर, एनीवन टेल मी, आर यू अंडरस्टँड द concept of categorical data or nominal data? Yes. भाग्यश्री जी, गोविंद सर, शिवकन्या, आवाज येतो का माझा? जाधव सर, नागेश सर, निकिता जी.
Yes sir, concept is clear.
हाँ सर, यस येतो का आवाज? यस. आकाश जी, आवाज येतो का माझा तुमच्याकडे? यस. यस, एनीवन टेल मी. अनम्यूट करू शकता का तुम्ही आता? नाही, किंवा चॅट बॉक्सचा तुम्ही वापर करू शकतो.
Unmute themselves.
हा, अनम्यूट करता येतील सर्वांना आता? यस. हेलो
सर सब म्यूटेड है सर.
हेलो. यस. यस, खान मॅडम, येतोय का आवाज? फिरदोस जी.
Yes sir, you are audible sir. आपका आवाज आ रहा है.
Hello everyone. Sir will be joined in 2 minutes.
Okay, ma'am.
Yes. आवाज तो आता? Anyone tell me?
Yes.
Yes.
Yes, sir.
खान मॅडम तुम्ही मला को-होस्ट करता का? होस्ट सायन्स कॉलेज नांदेड आहे. होस्ट केल्याशिवाय हे मला शेअर करता येणार नाही.
Hello, sir.
सर को-होस्ट करायला सांगा मला कोणाला तरी. कोण आहे सायन्स कॉलेज? ओके ओके. यस. खान जी आप कर सकते हैं क्या मुझे को-होस्ट?
नहीं सर.
हाँ हाँ होस्ट वो है सायन्स कॉलेज नांदेड जो है वो होस्ट.
हाँ सर, वो गणेश
हाँ,
गाठोडे
ओके,
सर
ओके,
करेंगे आप.
ओके, ओके. हाँ मी लावलं मराठी सरांना फोन. 1-2 मिनिट वाट पाहूया. ठीक आहे. तो पण आपण डिस्कशन करूया. तुमचे काही प्रश्न आहेत का? एनी डिफिकल्टी? निखिल जी प्रश्न काहीच नाहीत. प्रश्न नाहीत हाच प्रश्न आहे. येस ओके. ओव्हर झालेला आहे. येस स्क्रीन दिसते का ही?
Yes, sir.
आपण पाहत होतो categorical data, nominal data. What is the concept of categorical data? What is concept of nominal measurement scales, and which time researcher use nominal measurement scales? Generally, nominal measurement scales means very norms and words are used in a measurement process. That scale is considered as a nominal measurement scales, and generally qualitative research scholars are used in a nominal measurement skills. Means linguistic research student, language research student, they can use a nominal measurement skills using a measurement skills in research process, isn't it? And categorical type of data or numerical data, both type of data, you have to use a different measurement skills in each and every research process measuring a research data. Third one is a 1 minute. Yes, ordinal measurement scales, ordinal research data means whatever may be ordinal research data that was already words are used in you are measuring a research process. That time researcher use a ordinal measurement scales. Means longitudinal research information, you have to analyze some area, some aspects, some different words used in a frequently, like a primary, middle, secondary, higher education. That type of words we have to use in a ordinally. Means after one cycle, second cycle, after second cycle, third cycle, that type of information you have to use a ordinal measurement scales. Means one stata depending on the another stata depending on the third layer of research information. Means each and every factors, each and every layer depending on the each that type of research data you want to create, you want to generate, you want to collect in your research process. That time researcher use a ordinal measurement scales or ordinal type of research data, isn't it? Suppose in chemical sciences, means 30%, 40%, 20%, that type of you have to scales, you want to use in a your data analysis or your data measuring. That is called a ordinal measurement scales. Ordinal means you have to use a verb or words or different category words at a time. You want to use a measuring a purpose of your research data. That is considered a ordinal measurement scales. म्हणजे काय, खंडित शलाका त्याला असं म्हणतात. खंडित शलाका जे आहेत शब्दांमध्येच मानली जाते किंवा शब्दांमध्येच मापन केलं जातं आणि हे शब्द एकमेकावर आधारित असतात. अशा प्रकारच्या माहितीला आपण खंडित शलाका, मापन शलाका असं म्हणतो. येस. And next type of research data, that is numerical measurement scales or numerical research data that consider as a quantitative research data. Qualitative not. Categorical data that consider a qualitative. Means the main purpose of categorical data to measure the quality, to measure the errors in quality, and numerical data to measure the quantity, the impact of one data to another data. That time researcher use a numerical data measurements or numerical or quantitative research data measurements. Means direct research data or continuous research data, means whatever may be that measurements you want to use in a that particular research data. That data considered as a numerical data. In numerical data, these are the two measurement scales generally used in each and every research process. Like interval measurement scales. What is concept of interval measurement scales? Which time researcher use interval measurement scales? What was the characteristics of interval measurement scales? Interval measurement scales means whatever may be your research data divided into two sectors, equal sectors. Means 0 is absolutely considered. That type of research data generally used in a interval measurement scales, isn't it? Means like thermometers, whatever may be. Means 0, -5, +5, -10, +10. That type of research information generally researcher use interval measurement scales. Means these are the some measurements parameters used in a research data. Nature of your research data. म्हणजे माहितीचं मापन या प्रकारे केलं जात असतं. म्हणजे एक वेळेस शॉर्ट नोटला सुद्धा आलेला आहे. व्हॉट इज कन्सेप्ट ऑफ इंटरवल मेजरमेंट स्किल्स. आणि सेटला पण एक 2013 ला प्रश्न आलेला आहे की विच मेजरमेंट स्किल्स जनरली यूज इन अ एक्सपेरिमेंटल रिसर्च डिझाईन. प्रायोगिक अभिकल्पामध्ये कोणत्या मापन शलाकांचा वापर केला जातो. अशा प्रकारचा जो आहे एक. यस, एनी क्वेश्चन.
स्लाइड चेंज नहीं हो रही है सर, स्लाइड नंबर 4 चल रही है.
यस, अभी चेंज हो गई.
यस सर.
मैं आपको स्लाइड आपके ग्रुप में जो मराठी सर है वो भेजेंगे. यस, नाउ विजिबल दिसते का आता.
Yes, sir, visible.
ओके. हे जे आहे इंटरवल मेजरमेंट स्केल्स, which time researcher use interval measurement scale, what was the norms and standards of interval measurements? Generally in thermometer 0 is considered as a absolutely or silently, -5 +5 +10 -10, that type of research information you want to measure through a interval measurement scales. I mean that type of research information you want to create means your research information is numerical research information and divided into two different sectors. 0 is central. That time researcher use a interval measurement scales, isn't it? Means 0 that considered as a absolutely in between. Means in ratio measurement scale, that was the fourth parameters or fourth measurement scales generally used in a numerical research data. Means what was the ratio, what was the 1:2, 1:3, 1:4, that type of the percentage of one variables to another variables comparison, that type of comparison comparative study or comparative hypothetical study. That time a researcher use a ratio measurement scales or ratio data. म्हणजे काय तुलनात्मक अभ्यास करण्यासाठी किंवा जसं आता मेल अँड फिमेल रेशो इन नांदेड सिटी इन नांदेड डिस्ट्रिक्ट, that time researcher use a measurement scale, ratio measurement scales, isn't it? Means comparative study of one variables to another variables. Suppose any two crop, you comparative study of a farmer survey, comparative survey of any one or two crops. That time researcher use a ratio measurement scales, means 0, 5, 10, 15, means 5% gap, or 1:2, 1:3, means teacher student ratio, isn't it? Like demand and supply ratio. That time researcher use a ratio measurement scales analysis. These are the four measurement scales generally used in a measurements of research data. Means that four research measurement scales divided into two categories. First one is categorical research data, numerical research data. Another first category categorical research data, nominals and ordinal measurement scales generally used. These are the two measurement scales generally used in a categorical research data. And another type of research data, numerical research data, another ratio measurement scales and interval measurement scale generally used in a measurement scales. These are the two measurement scales generally used. Yes, these are. हे जे आहे ह्याच्यावर एक शॉर्ट नोट येणार आहे. हे चार्ट फक्त लक्षात ठेवायचा. Concept of categorical measurement scales, concept of numerical measurement scales, nominal measurement scale, ordinal measurement scales generally used in categorical type of research data, interval measurement scales and ratio measurement scale. These are the nominal or numerical measurements scales generally used in a data measuring. Yes, ह्याच्यामध्ये काय अडचण आहे का? चलायचं पुढे. म्हणजे 5 मार्क इथे कव्हर झाले. आता लॉंग क्वेश्चनचे आता आपण एक पाहणार आहोत. Yes, ratio measurement scale झालेला आहे. Yes, primary source of data and secondary source of data हा पण लॉंग शॉर्ट नोटला विचारलेला आहे. What was the concept of primary sources of research data and secondary sources of research data? Means generally primary research data considered as a origin of your research data, the main source of your research data, that considered as a primary source of research data or primary data collection. म्हणजे जे काय primary research data, which time researcher consider a primary source of research data, you have to consider a main source of your research data. That time researcher consider a primary research data. म्हणजे मुख्य स्रोतातून आपल्याला मिळवलेला आहे ती माहिती किंवा त्रिकालबाधित सत्य माहिती जी आहे. जसं आता गव्हर्नमेंट GR, उद्या सुट्टी आहे असं जर मी म्हटलं तर ही सेकंडरी डेटा झाला. जर नांदेड जिल्ह्याला उद्या सुट्टी आहे असं जर मी म्हटलं, जर नांदेडच्या कलेक्टरनं त्याच्या सहीचं परिपत्रक काढलेलं आहे, दॅट कन्सिडर ॲज अ प्रायमरी सोर्स, isn't it? That considered as a primary source. The main source of research information considered as a primary source. जेवढे काही इन्स्ट्रुमेंट मध्ये आलेले रिझल्ट आहेत किंवा त्याला काय म्हणतात रीडिंग आहेत, that consider as a primary source of data. And generally that primary source of consider data that consider two segments, means qualitative research data and quantitative research data, means verbal research data or numerical research data, isn't it? And these are the some research data collection tools, means research tools, means qualitative research tools and quantitative research tools. These are the two type of research tools researcher use in a various research process. म्हणजे गुणात्मक माहिती आणि संख्यात्मक माहिती असे माहिती संकलनाचे दोन प्रकार आहेत. Research data collections or research data tools divided into two types. One is qualitative research tools, another is a quantitative research tools. And main theme is a qualitative research data and quantitative research data researcher transfer in one form to another form. एकातून दुसऱ्यामध्ये आपल्याला परिवर्तन करता येतं, परंतु दोन प्रकारामध्ये ही माहिती आपल्याला गोळा करावी लागते. फर्स्ट वन इज क्वालिटेटिव्ह रिसर्च टूल्स अँड क्वालिटेटिव्ह रिसर्च टूल्स म्हणजे काय, गुणात्मक माहिती जी आहेत हे जे आहेत क्वालिटेटिव्ह रिसर्च डेटा रिसर्च टूल्स, मीन्स ऑनलाईन ॲज पर द कन्सर्न ऑफ ऑनलाईन रिसर्च टूल्स, ऑनलाईन फॉर्म असतील, डिप इंटरव्ह्यू असेल, ऑनलाईन इंटरव्ह्यू असेल, ग्रुप डिस्कशन असेल, वेब सर्वे असेल, ऑनलाईन कम्युनिकेशन असेल, दिस ऑल कन्सिडर ॲज अ क्वालिटेटिव्ह रिसर्च टूल्स. Qualitative research tools. Qualitative methods are presentative numerical requirement. 1 minute. Yes. Or quantitative research tools. Quantitative, which time researcher use a quantitative research tools? These are the some process of research data collections. Yes, we discussed regarding the research tools, qualitative research tools and quantitative research tools. Two types of research tools. First one is interview considered as a qualitative as well as quantitative research tools. Both the type of research information you have to collect through interviews, means numerical type of research data or verbal research data. Both categorical research data or numerical research data you have to collect through a interview. Interview is a very easily use research tool for a purpose of research data collection. Generally humanities research scholars or interdisciplinary research scholars and commerce and management research scholars was use interview is a popular research tools. Means what is concept of interview, what is the definition of interview, what was the characteristics of interview, what was the merits and demerits of interview. म्हणजे तुम्हाला ज्या शब्दामध्ये तुम्हाला लिहिता येईल त्या शब्दामध्ये तुम्हाला लिहू शकता तुम्ही. द कन्सेप्ट ऑफ इंटरव्ह्यू तो फेस टू फेस और डिस्टन्स और ऑनलाइन इंटरव्यू, ऑफलाइन इंटरव्यू, डायरेक्ट इंटरव्यू, इनडायरेक्ट इंटरव्यू, फोकस इंटरव्यू, टाइप इंटरव्यू, अनशेड्यूल्ड इंटरव्यू, शेड्यूल्ड इंटरव्यू. These are the some types of interview. Generally the interaction between two persons that considered as a interview. The interaction between or conversion between two or more than two persons, two individual persons or more than two individual persons, that process is considered as a interview. Means as per as the concern of the purpose of collection of relevant information or satisfactory research purpose or satisfactory research data information, that process is considered as a research interview or purpose of research interview. Means whatever may be two or more than two persons conversion or interactions that consider as a interview. The main purpose, the focus of that interaction is to collect some information regarding your research purpose. म्हणजे काहीतरी माहिती मिळवण्याच्या अनुषंगाने जे काही संवाद साधला जातो त्या संवादाला काय म्हणायचं आपण, मुलाखत असं म्हणतो. आणि मुलाखत ही दोन्ही प्रकारची असते. गुणात्मक माहिती आणि संख्यात्मक माहिती ही मुलाखतीमधनं आपल्याला मिळवली जात असते. स्ट्रक्चर्ड इंटरव्ह्यू, अनस्ट्रक्चर्ड इंटरव्ह्यू, फोकस इंटरव्ह्यू और नॉन फोकस इंटरव्ह्यू, फॉर्मल इंटरव्ह्यू, दीज आर द सम टाइप्स ऑफ इंटरव्ह्यू. म्हणजे काय, मुलाखतीचे हे खालील 5 प्रकार आपल्याला म्हणता येतात. म्हणजे काय, संरचित मुलाखत, असंरचित मुलाखत, खुली मुलाखत, विषयांचित मुलाखत किंवा मग माहितीपर मुलाखत, ही जी आहेत मुलाखतीचे हे काय 5 प्रकार मानले जात असतात. आणि दीज आर द सम कॅरेक्टरिस्टिक्स ऑफ interview as a research tools. These are the some merits and demerits of. 1 minute. Screen दिसते का? Yes, anyone tell me?
No, sir.
Screen दिसत नाही का? Okay, 1 minute. Yes, now is it visible?
Yes, sir.
हे सोपं आहे. असं काही नाही, तुम्ही जनरल जरी ह्याच्यात लिहिलं प्रत्येकाचे कॅरेक्टरिस्टिक वेगवेगळे येऊ शकतात. इच अँड एव्हरी रिसर्च स्कॉलर दॅट रिसर्च टूल्स यूज अ डिफरंट वे, different techniques and different character you have to use. The purpose of interview may be a different, but that purpose you have to consider as per as the concern of a researcher views. Means aims and objectives of your research tools, that was different. Format of your research tools, that was different. Person to person, area wise, your research area, means research tools format are different, research tools characteristics are different, research tools steps are different. That depend upon the person to person, area wise researcher. Research tools, sometimes research tools developed, self-developed research tools, or sometimes standard research tools researcher use in your research process. Means standardized research tools or non-standardized research tools. These are the two type of research tools generally used in each and every research process. Standardized research tools means already so many agencies standardize that format. That research tools is considered as a standardized research tools. Or self-developed research tools means researcher developed research tools, isn't it? That is called non-standardized research tools. These are the some characteristics of interview as a research tools. Means whatever may be the interaction between two or more than two persons that consider as a interview. The characteristics of interview means causal factors or determinations to find out the attitude, to measure the attitude, to measure the conversion of origin, the conversion of that information, the origin of that problem, to sources of that information. You have to find out the through a research tools. Interview as a research tools. Appropriate ideas you have to collect. Whatever may be appropriate information, relevant information, true information, you have to collect through a research interview. Means research interview is a very easily literate persons or non-literate person, both type of audience, both type of research population, very useful research tools. Interview as a research tools, or each and every age limits means small child, middle child, upper, older human beings, that all area, all types of research respondents, researcher can use a interview as a research tool. That was the characteristics of a research tools. Means it can make a cross questionnaires possible. Means you have to check the that is a real response or artificial response or truth response, or sometimes the validity of that research information. A researcher check through a research interview, that research tools. Means investigations, what was the truthness of that investigations, what was the purpose of that investigations that was helpful for a research purpose. That is a basic characteristics of research tools as a research interview. Means confidentiality or deftness of that research tools, interview deftness, interview confidentiality, subjectivity, validity. These are the some characteristics of interview as a research tools. Or flexibility. Flexibility is a most ideal characteristics of interview as a research tools. Means difficulty level you can change at the time of response collect. Yes. Any question? सारिका जी आहे काय प्रश्न?
सर आय एम ड्राईव्ह मोड चुकून ऑन झालेला आहे. एक मिनिट.
ओके ओके ओके. समजतं ना तुम्हाला? तुम्ही आजच क्लास जॉईन केला आता अगोदरचे 2-3 क्लास मध्ये नाव दिसलं नाही तुमचं सारिकाजी. ठीक आहे. Flexibility, that was the most important characteristics of interview or whatever may be a truthness, judgment through a interview. That was the ideal characteristics of interview. Respondents modification or answer modification, answer artificialness. You have to find out that was the characteristics of or holisticity or experiences or case studies or clinical studies. Research scholars can use that interview as a research tools. These are the some merits of research tools. Interview as a research tools means direct research, direct information you have to collect for the purpose of research process or for the purpose of research validity. The truthness of your research information, that is a merits, depthness of information as per the concern of various types of depthness. You researcher can create the depthness or various angles of that research information, the knowledge of the past, future, and present, all types of knowledge you want to create through a research interview as a research tools. Means knowledge specifications or features of the knowledge. That was the merits. That was the merits of research tools. Interview as a research tools means enhancements or engagement or whatever may be. The alternative net of flexibility, that was the merits of some research tools or possibilities of the observation or scientific observation, behavioral observations or psychological process or possibilities or behavioral possibilities you can judge, you can measure through interviews. Means knowledge is a historical background or emotional background cases. You can that is the merits of interview as a research tools. Or both type of research data you want to create through a research tool or interview as a research tools, or examinations of the knowledge data or examination of the measurements of the that experiences or knowledge or real data. That is the merits of that research tools. म्हणजे मुलाखतीचे फायदे एक 7-8 फायदे तुम्हाला जे वाटले ते लिहायचे, असं काही नाही हेच लिहावे असे काही तुम्ही लिहिते ते बरोबरच असतात. रिसर्च मध्ये काय ते सर्व बरोबरच असतं. प्रत्येकाचं रिसर्च मेथड ही बरोबरच असते. रिसर्च मध्ये चूक काहीच नसतं. त्याच्यामुळे काय तुम्हाला जे वाटले ते लिहायचे. These are the some demerits of research tools as a interview. म्हणजे मुलाखतीच्या काय ह्या त्रुटी आहेत. म्हणजे कृत्रिम प्रतिसाद देऊ शकतात, इरिलेव्हंट रिसर्च इन्फॉर्मेशन, इरिलेव्हंट प्रेडिक्टेशन, इरिलेव्हंट क्वेश्चन्स, किंवा मग जे काही लो लेवल ऑफ इंटेलिजन्स, इमोशनल इंटेलिजन्स और अंडरस्टँडिंग, डिसेबल स्टुडंट्स, that time disable population, that time researcher not use research tools as interviews. म्हणजे जे काही मूकबधिर आहेत, कर्णबधिर आहेत किंवा बाकीच्या प्रकारचे जे काही विशेष विकलांग आहेत अशा ठिकाणी हे जे आहेत हे इंटरव्ह्यू आपल्याला वापरता येत नाही. These are the some demerits किंवा incomplete information, complete information, irrelevantness of research information. These are the some demerits of interview as a research tools. And what was the difference between questionnaires and interviews? These are research tools, qualitative research tools and quantitative research tools. This questionnaire as used as a numerical types of research data, and interview as a qualitative research data. Questionnaires collect a quantitative research data and interview collect a qualitative research data. What was the difference between qualitative research tools and quantitative research tools? That type of questions asked in previous examinations. Means these are the some angles the difference between qualitative research data and quantitative research data tools. म्हणजे गुणात्मक माहिती आणि माहितीचे साधन आणि संख्यात्मक माहितीचे साधनामध्ये हा फरक आहे आणि फरकावर एक प्रश्न लॉंग प्रश्न विचारलेला आहे. Isn't it? Means what was the difference between interviews and questionnaires? These are the difference between interviews and questionnaires. Means no face-to-face contact between two or more than two persons, or knowledge level, difficulty level, topic of difficulty level, interactiveness of difficulty level, to writing format only, not confidentially maintained, or not preserved, not gathered indirectly or directly, or data is gathered directly in first way, indirectly in second way, the data can collect directly, face to face contact in interview or interpretation was be different. Means skillful interviews is needed, means skill is essential in that another tools and techniques, and first tools and techniques is not essential in questionnaires. In interview skills is various types of skills is essential, or flexibility or not flexibility. In second way, flexibility is used, means various some confidential level, confidential informations you can preserve in second types of research tools. That confidential levels are in written form in first types of research tools, means written or information, both type of research information you have to collect. These are the some types of research tools. Interview as a research tool, semi-structured, structured, unstructured. These are the types of research tools. Yes. हे झालं इंटरव्ह्यूचं. आता आहे क्वेश्चनरीज. क्वेश्चनरी ॲज अ रिसर्च टूल्स. प्रश्नावली हे एक संशोधन साधन आहे. What is the definition of questionnaires? What is the characteristics of questionnaires? What is the merits and demerits of questionnaires? And what was the types of questionnaires generally used in a various research process? Isn't it? Means list of questions related to your topic, that is considered as a questionnaires. म्हणजे प्रश्नांची शृंखलाबद्ध रचना ती विषयाशी निगडित असलेल्या आराखड्याला आपण प्रश्नपत्रिका असं म्हणतो. जी अगोदर आपण बघितली दॅट कन्सिडर ॲज अ क्वेश्चनाराइज. ही अगोदरची प्रश्नपत्रिकाची बघितली दॅट कन्सिडर ॲज अ क्वेश्चनाराइज. म्हणजे नियमबद्ध किंवा शृंखलाबद्ध प्रश्नांच्या रचनेला प्रश्नपत्रिका असं म्हणतात, अशी त्याची व्याख्या लिहून टाकायची. It is a list of questions related to your topic that consider as a questionnaires. These are the definition of your research tools as a questionnaires. These are the some characteristics of research tools. Questionnaire as a research tools means structured questionnaires, unstructured questionnaires, objective questionnaires, unobjective questionnaires, open-ended questionnaires, symbolic questionnaires, non-symbolic questionnaires. These are the some types of research tools as a research tools as a questionnaires. म्हणजे बंदिस्त प्रश्नपत्रिका असतील, मुक्त प्रश्नपत्रिका असतील, संचित प्रश्नपत्रिका असतील, अंशतः रचित प्रश्नपत्रिका असतील, किंवा काही सिम्बॉलिक असतील, जसं आता चौथीच्या वगैरे आहेत, वेन डायग्राम वगैरे एमपीसीला वगैरे विचारतात. That is a symbolic questionnaires. These are the some types of questionnaires generally used in a research process. Means questionnaires used as a research tools as in various research information. You have to collect through a research questionnaire as a research tools. Within few seconds, within few time, we have to collect a lot of response. That is a very popular research tools. कमीत कमी काय म्हणतात वेळेमध्ये जास्तीत जास्त माहिती संकलित करण्यासाठी प्रश्नपत्रिका हे एक चांगलं साधन आहे. These are the some characteristics of research tools as a questionnaire as a research tools. Means details with importance and significantness of your research topic, relevantness of your research topic. These are the some characteristics. Clarity, questionnaires, difficulty level, question itself, letter format. These are the some characteristics of sequence, deafness characteristics, and validity and reliability of your questionnaires, and difficulty levels of questionnaires, sequence of your questionnaires. That was the characteristics of question arise as a research tools. Research skill means how to develop your questions, how to obtain resources report or recorded research report. Means recorded research respondent responses. That is a one basic characteristics of question arise. म्हणजे लिखित स्वरूपाचा प्रतिसाद तुम्हाला नोंदवता येतं, हे सुद्धा प्रश्नावलीचं एक मुख्य वैशिष्ट्य आहे. और पॉसिबिलिटीज जी आहेत ती हे जे आहेत हे सुद्धा काय इसेन्शियल डेटा. यू हॅव टू गेट लॉट ऑफ इन्फॉर्मेशन, लॉंग इन्फॉर्मेशन, क्रॉस इन्फॉर्मेशन, longitudinal type of research information and cross sectional type of research information you collect through a questionnaire as a research tools. And direct collections or indirect collections, direct question sequence, negative attitudes, positive attitudes, adjustments, thinking level, writing level, that ability you have to check. That is the characteristics of research tools as a questionnaire as a research tools. And these are the some merits and demerits of questionnaire as a research tools. टाइम सेव्हिंग प्रोसेस आहे, लार्ज अमाउंट्स ऑफ रिसर्च रिस्पॉन्सेस कलेक्ट होतात, रेकॉर्डेड आहे, स्पेसिफिक रिस्पॉन्स यू हॅव टू कलेक्ट, रिलेव्हंट रेकॉर्ड यू हॅव टू मेंटेन अगेन अँड अगेन. यू कॅन चेक थ्रू अ रिसर्च टूल. दॅट वॉज द डिमेरिट्स ऑफ युअर रिसर्च टूल्स. हे प्रश्नावलीचे काय हे मर्यादा आहेत. म्हणजे काय हे डिसेबिलिटी स्टुडंट दॅट टाइम रिसर्चर नॉट यूज questionnaire as research tools, lack of personal contact, whatever may be writing skills, writing format, analytical speaking, measurements, evaluation process that affecting on research data. Means various types of persons you have to create various types of measurements in your research data. Now these are the some techniques generally used in a questionnaire as a research data. Means recorded data, digital recorded, non-digital recorded. You can use in interview or questionnaire as a research tool. Both type of research tools, qualitative research tools and quantitative research tools, you have to use a recorded information. Means similarly recorded or digital recorded you can use. Means focus group. These are the generally used in a quick respondence. You want to solve a quick respondence through a population. That time researcher use a focus group. Means 10 to 15 students participate in that focus group, and as per the one task given through various types of idea you want to collect through a interview. That time focus group you can use in a qualitative as per as the quantitative research tools. Okay. Yes, any difficulty, any questions? आहे काय अडचण? म्हणजे सेम हीच पोझिशन तुमची झाली असेल, म्हणजे काही म्हणजे लक्ष आहे का नाही माहित नाही. येस, एनी क्वेश्चन, एनी डिफिकल्टी? असेल आपण थांबूया. जाधव PS क्लिअर सर. आवाज येतोय का सर्वांचा? अनम्यूट करू शकता तुम्ही? येस. एनी डिफिकल्टी, एनी क्वेश्चन? आहेत काय प्रश्न? खानजी, मिर्जाजी, गोविंद भोसले? यस. आहे काय प्रश्न? 1-2 मिनिट. ठीक आहे, आपण इथे थांबूयात. धन्यवाद सर्वांचे.
Okay, sir.
Thank you.
On the behalf of Research Center and all the research scholar, I am very thankful to you, sir, for sharing this very informative information or data with us, sir. Thank you so much, sir.
Okay, thank you.
Ganesh, sir. गणेश सर मीटिंग लीव एंड करिए सर. गणेश सर.